out to fathers and siblings and collect or construct family-based designs, I think that will be very beneficial in epigenetics.
Yes, Dr. Grove.
I'd like to actually ask a question of the geneticists and the epigeneticists. Do we have the knowledge and the computing power right now? And this is actually based on what Rob had brought up yesterday, was that, you know, it used to be that methylation meant you were suppressing and demethylated meant, well, we know that that's not the case. So by just simply looking at a methylation pattern, you can no longer predict whether something will be suppressed or enhanced or whatever. So do we have the knowledge? And when we have that complexity built in, when we tier this data, do we have the computing power to actually predict what the likely biological outcome would be?
I would say that yes to both. That I mean, as evidenced by the recent Nature paper, Kundaji et al. That summarizes the findings of the Roadmap Epigenome Project. I mean, that's an indication of the state of the art, really, in the ability to link epigenome-wide annotation of various marks at different levels, you know, in terms of not only DNA methylation, but also various histone modifications, et cetera, with gene expression in those associated samples. And so really the question now is, do we have the, I would say, the political will to make the investment that's necessary to understand, you know, questions such as inter-individual variation in these mechanisms and developmental changes, you know, really digging into cell type and tissue specificity, all these types of things. But yeah, certainly, and sequencing costs just continue to decline. So more and more now, the real expense of doing these studies is going to be data management and data analysis.
Yeah, I think the computational power is there. The data is accruing undoubtedly with ENCODE and NIH roadmap, and I think it's just a case of beginning to knit it together and place that in the context, or use the population-based data in light of the huge array of data on the epigenomic landscape that's being generated.
Dr. Barcarelli?
Yeah, I would like just another option for that. I mean, obviously the easiest thing to do is to measure RNA or protein expression in your studies. Another option, if you don't have the sample, so you don't have the financial part of your study to pay for it, is actually to go online, and there are many databases now in which you can get DNA methylation and RNA expression. Perhaps you will be lucky also to find in the tissue you're interested in. Another thing that I want to point out, though, is that many times when we do that, we don't find the association between DNA methylation and gene expression. And the reason is that DNA methylation is only one of the layers that control gene expression. And if you look at histone modification, that would be the same. And here I also need to keep in mind the concept that has been evolving in the past few years of genes that are poised to be activated. And this is probably something we need to keep in mind, that there might be information of these genes that are poised to be activated that will be more easily activated, and that could be also interesting information, especially in the framework of longitudinal studies.
Okay, so
just to add a couple things on what has just been said, I think we are, like, the sequencing cost is getting lower. We're getting more and more computer capacities to analyze the data and manage the data. I think the next challenges will be how to interpret that data in the multiple layers, just like Andrea mentioned. We're gonna need teams of both, like, geneticists, physiologists, and then people who want to understand the overall biology and what it means. But also, it might also not just differ on the tissue, but also in the context. We found some signals in pregnancy for the same biomarkers that we don't find outside of pregnancy for genetic association. So there's going to be more than one layer to interpret.
Okay, so in the interest of time here, I'm going to hold those further comments, and we can certainly continue in the next discussion, but I'm going to introduce Dr. Judith Hall who is going to be our facilitator for the opportunities and challenges in epigenetics research.